Hi, I'm Pat Lorton. I'm the CTO of Schrodinger, a company many of you may not have heard of, working on problems you also may not have heard of. But I'm excited to talk to you about both today. I think there's a lot of interesting stuff in there, even for people who aren't from the field. So, who is Schrodinger? What do you do? Those are going to be the first questions. We're an almost 30-year-old company that is committed both to fundamental research and applying that research to accelerate drug discovery and materials design. Our approach to accelerating drug discovery is to simulate chemistry on the computer. I imagine that's very fascinating to a lot of people in here. And it allows us to make predictions about how drugs will work in the human body without ever having to create those drugs in a lab and test them until much later in the process. I won't have enough time today to talk about materials design, but there's also a lot of really fascinating stuff there, from OLED design to perfumes, just tons of fascinating chemistry we're able to simulate in those fields. I will start with a quick walkthrough of the agenda. One thing I really want to state up front is I'm trying to present in a way, realizing that most people, this field is completely novel and you don't know anything about it. So you don't have to know about computational chemistry or even chemistry for that matter. I'm going to try to break it down into the core problems of computers, but I will also give some background because otherwise it's just not going to be an interesting presentation. So first, we'll start with, why is drug discovery so hard or expensive? You've probably read about this. Newspapers talk about it all the time when this new drug comes out and it costs $200,000 for a cure for something, and everyone says, how can you charge that much? They'll reference that it costs billions of dollars to discover the drug. Why? Why does it cost billions of dollars? So I'll start with that. And then I'll go to computational chemistry, which is a field that is really more than 50 years old at this point, but is seeing a renaissance with modern compute. I'll talk about Schrodinger's solution, which uses the cutting edge computational chemistry to solve pharmaceutical problems. And then from there, after that background, I'll go into the compute requirements, the real meat and potatoes, and we'll figure out what GCP is doing that's driving our science and driving drug discovery faster. In that part, talking about how GCP is speeding up Schrodinger, I'll go through several things. I'm not going to read them off here, but there are many ways that GCP is really being helpful to us as a company and as the field in a whole. So let's jump in with this big crazy number. There are more than 10 to 60 potential drug-like molecules. Some estimates go all the way up to 10 to the 100th or 10 to the 120th. That is just an astronomically big number. By some estimates, it's more potential drug molecules than there are atoms in the entire universe, which is hard to even fathom. And we need to find the best one for each disease. That's a little bit strong. It doesn't have to be the absolute best one, but we have to find a really, really good one for each disease. And when you're looking at that many potential drug-like molecules, it's an incredibly daunting task. It's not like a needle in a haystack. It is not even an atom in a haystack. It's smaller than an electron in a haystack. That's how small of a thing we're looking for when we're trying to find a good drug. So why is drug discovery as a whole so hard? I just got into the nearly infinite chemical space. That's one of the most daunting tasks in preclinical drug discovery, when we're looking for the right drug before we take it into human clinical testing. Accurate evaluation is really hard. Even if you make a guess, if I just randomly am able to pick the magical molecule out of my head and make a perfect guess, synthesizing that in a lab costs somewhere from hundreds to thousands, and in some extreme cases, even hundreds of thousands of dollars to produce that molecule so you can test it. So figuring out how to test ideas faster and cheaper can really bring down costs. When you're saying, oh, let's try to do it computationally, the problems are extremely computationally hard. So it's a constant negotiation between accuracy, which we may never be able to compute perfectly, or something really, really fast that might have so low accuracy you can't really make decisions off of it. So that's the pull and the push in computational work. And then if you do all of that right, if all your preclinical work goes perfectly, biology is also really hard. Biology is where drug design projects start, because you have to know what biological target you're going after. And it's also where they end, because when they're going into the clinic, you're really testing whether the chemistry works well in the human biology. And you had theories going into it, but your theories are really just a hypothesis until you've done large-scale clinical trial testing. And the vast majority of programs actually fail during clinical testing. So even if you do everything perfect chemically, which is really expensive and hard, you still stand a decent chance of failing because the fundamental biology was wrong. So that's why it's so dang hard and expensive. So what is computational chemistry? I won't bore you by reading the entire thing, and you've probably guessed by the name. It's using computers to make chemistry predictions. It's an incredibly hard problem. Without using numerical analysis or clever estimations, many chemistry problems are more complex to solve than the hardest encryption algorithms. We're talking, it is easier to crack the hardest encryption algorithm than it would be to get the exact accurate answer for a chemical question, which is really, really daunting. So we have to try to estimate. The main goal of this field actually is to try to get as accurate a solution as possible with as fast of estimations as possible. Why is it so hard? Well, again, we're going back to nearly infinite chemical space. The more predictions than atoms in the universe, if you take that to the next step when you're thinking about the infinite space, that means it is, literally impossible to store. If you somehow could calculate the perfect value on every single one of those, you couldn't even store them because there's not enough atoms in the universe to put that data anywhere. So you have to be really, really clever in what you sample and how you store. Accurate evaluation is really hard. What I was mentioning a second ago about how it can be more difficult than encryption algorithm. These are NP hard problems and very hard NP hard problems. Quantum computers may one day crack some of them, but there are a lot of problems that are just not going to be amenable to quantum computers ever, just because of the nature of the data and things like that. Then you start talking about how big, if it's that hard for a simple water molecule, like I mentioned here, how big it is. Proteins are actually huge. So if it's so hard that quantum computers still can't solve for a water molecule, or the fastest supercomputers are having problems getting valid values for a water molecule, we're talking hundreds of thousands of atoms, orders of magnitude larger than water, that we need to be able to accurately predict to make a drug prediction. Huge amounts of science, 50 years, have gone into accurately estimating relatively cheaply. And then the compute is massive and uneven. So that is something that the cloud has really helped a lot, because in certain phases of a drug discovery project, you might want 50,000 GPUs to be working for a day or two to get you an answer quickly so you can drive decisions in the lab. But then you'll have a 3-week synthesis cycle in the lab where you just don't need those GPUs at all. And if you're at a big pharmaceutical company, you might be able to balance it by having so many projects vying for that time. But a huge amount of fundamental drug research is done at smaller biotechs that don't have that advantage of dozens of active projects going on. So I'm going to stop from kind of the more compute intensive description and do a really quick science break. I know most of you won't know chemistry, so this just looks like 3 random pictures on the screen. So let's pretend that our brains are neural networks, and try to do some pattern recognition here. Solubility is one of the most important properties in drug design. We've spent a huge amount of resources trying to accurately predict solubility. Solubility, just as a reminder, is simply when you drop a molecule, or I'm sorry, when you drop a drug in water, will it actually dissolve into the water? If it doesn't dissolve into the water, if it just sits there, then what you have is extremely expensive brick dust, because it will just sit in your system and go completely through you without your body ever absorbing it and using the drug. So you have to find something that's soluble. So looking at it from an image recognition neural network, these are 2D representations of 3 different structures. So let's try to guess the third structure based on 2 where we already know the answer. So the left one has mediocre solubility. It'll break down in water a bit, but maybe not enough for a drug to be effective. The far right one has no insolubility. It'll end up as chalk in the bottom of your water. And the middle one kind of looks like something between the 2. So it's really hard to guess. Maybe it has a little better solubility than the left one. Maybe it has in between. But from this data, there's just not much you can do. And while you can train on huge, huge data sets, there are some inherent properties that make it so that it's really, really local. And there are these things called activity cliffs, where changes happen very dramatically in chemistry that make active learning and machine learning very difficult to solve problems like this. So you might have guessed that I was going to give you a surprise. This compound in the middle is actually very, very soluble. You drop it in water and it comes apart, and you can very effectively deliver a drug with this type of solubility. So why? What's the difference here? So the insoluble compound, to understand it, you have to go to 3D. This is taking that insoluble compound, rendering it in 3D like it is in real life, and then packing it into a crystal, similar to how water packs into ice crystals. you can see all of those blue connections there between the different copies of the molecule. Those blue connections are moderately energetically favorable. So if you have a bunch of blue connections, that might be enough to hold something together, maybe not forever, but for a long time. You can also see the yellow connections. Those are very, very energetically favorable. What that means is that with all those yellow and blue connections, this molecule really likes to hold on to itself. It packs really well together. And if it's packed together, it's not going to be tempted to go swimming with water when water comes around. It's going to hold on to itself. If you look at the moderately soluble molecule, you can kind of get an idea for why it is able to come apart a little bit. You'll see there are none of those yellow lines that were really, really strong. All you have is the blue lines, which is a weaker type of interaction. But there are enough of those blue lines that it still holds together quite well. So what you see when you take the other molecule, which we don't even have a good crystal structure packing here, is that you can't really pack it in a way where there's a lot of good energetic connections. There's many different ways you could pack it, but when it ends up that water comes around, there's so few interactions, and water is able to interact with it favorably that it just pulls it right out. This is really hard to predict with just 2D or 1D data. And honestly, we've run a bunch of ML because we're interested in machine learning a lot as well. And even when you take 3D data into consideration, if you're not actively looking at how the molecules can twist and turn and move with each other, the simple 3D snapshot is just not enough to predict this. This has been your science break, by the way. I won't try to teach you any more chemistry today, but I figured it'd be useful at some level to understand what type of properties we're talking about. Now, that property, solubility, it's pretty tough, as I was just talking about. It's actually relatively easy because solubility, at least, is only specific to the molecule. A lot of drug-like properties are how that drug molecule reacts with giant proteins or with other molecules that are much harder to predict. At least solubility, you're only thinking about that one molecule it's interacting with. But it's also only one of many, many properties we have to optimize. And all of those properties have to be good. If any of those properties are bad, it will kill a drug. And more evil than that is that not only are these properties typically not correlated at all, but some of them are even anti-correlated. So when you improve a property, it's guaranteed to make another property worse. So you're trying to find this tightrope between two properties where you can get it right inside the range of acceptability. This is long, laborious, tedious work. And when every guess you have about an improvement on the molecule requires weeks or months and tens of thousands of dollars of synthesis, it quickly adds up. You can start to understand with the vastness of molecules how hard this guesswork is and how expensive it is to try a theory. You can start to understand why drug discovery is so hard. I guess that's probably really the end of the science lesson here, but it is going to go to compute soon, I promise. So how is Schrodinger solving this? So we have a massive investment in fundamental research. That's something we've really focused on. Going all the way back. We were founded by Rich Friesner, who's still at Columbia in the chemistry department in Columbia, professor. And going all the way back to the beginning, we've really focused on scientists. We have over 100, I think actually it's over 150 PhD scientists at this point who all they do is try to figure out how these fundamental physics work on a chemical level and how we can encode that in our algorithms. The next step from the science is finding that perfect balance of accuracy and speed. And this is something both our scientists and a large engineering force work very regularly on. Sometimes that's as simple as speeding up our code. Sometimes it's moving things onto the GPU. But sometimes it's stepping back and saying, on a chemical level, what things can we take for granted that will allow us to simplify the algorithm? It wouldn't be robust to everything in the world, but for this problem, what can we simplify? So we do a lot of that. We do, luckily, scale embarrassingly parallel. Almost all of these calculations, since you're looking at one drug at a time, and you might want to look at batches of 10,000, that allows you to scale up really, really quickly, as long as you have the infrastructure to do so. But the last one then comes back around, which is if you're calculating information on billions or tens of billions or in the future hundreds of billions and trillions of compounds, how do you actually deal with all that data you're generating? And that's a big problem for us as well. And kind of how that works is on the very left phase, we're looking at billions. I think you can actually go up to 10 to the 10th at this point, where we're calculating very fast properties on CPUs for billions of compounds. We use some machine learning and some other simple algorithms to get that down to a small enough number that we want to push forward. And then we start these cycles where we improve the molecule, we make guesses, we improve our active learning. Humans will evaluate the designs, humans will synthesize an assay, and we'll slowly be improving these properties. The goal is to do this cycle as few times as possible. If you only had to do it once, it would be this dream world where you could go from an idea to a drug candidate going into the clinic, within weeks or a few months. That's the future dream. Right now, even in the best case scenario, you're still doing this cycle many times. So this compute requirements that I was referencing earlier, what do those look like? Well, we have massive scale for CPU. Maybe it's not massive scale for Google, but for most of us out there, 500,000 CPUs is a lot of CPUs to be using at once. Especially, I mean, that's more than 1,000 a person working at our company, to give you an idea of that scale. We also regularly need a diverse GPU. So the GPUs are actually the workhorses for our most important calculations. We have gotten relatively far down the filter of accuracy by the time we get to the GPU calculation. So we're doing it on a lot fewer molecules, which allows us to only maybe have 10,000 things running at once instead of 500,000 things running at once. That said, it's still a lot of work. We are lucky that it's relatively small input, and the final output is still relatively small in terms of its terabytes. If you were to keep track of all the data we generate along the way for those predictions, it's into the many, many petabytes of data. But luckily, we only really need to save some snapshots or final snapshots of these trajectories. We don't have to keep all the data together. So relatively small on the order of hundreds of megabytes or even gigabytes, and then only maybe terabytes out. So in the big sense of the cloud, pretty small. It's rich, dense data. It's that small because we want to analyze about everything. It's not like the first step is cleaning a bunch out. We really do want to look through pretty much all of that data. And then finally, how are we actually calculating things? We have very different requirements. Sometimes we need GPU node with only 12 gigs of memory, or even less, actually. I think we have some 6 gig instances. And sometimes we need 400 gigs on a GPU node. So the ability the GCP has to allow you to dynamically pick your node size is really, really powerful for us. So here is an example of a high memory T4 GPU node. So this is something that we used to have to build in-house. We actually originally deployed this application on an in-house server because you simply couldn't get something with a few GPUs and 400 gigs of memory. So I will go ahead and play this video. So what you're seeing here is we've loaded about 1.5 billion compounds into the GPU's memory as a subset. We have a secondary screening on the CPU, but allows us to keep a relatively easy microservice, if you will, that is looking over 1 billion synthesizable compounds. And what we're doing is we're saying those compounds on the right are searching libraries that you can just purchase them off the shelf. You don't have to synthesize them, which is a huge advantage in early drug design. And we're saying, okay, I'm drawing my IDs on the left. Find the closest thing I can just purchase off the shelf so we can kind of guess and experiment with our ideas without having to invest in custom synthesis, which is very expensive. So how are our strategies for executing the big backends? Not that microservice, but when we're calculating properties for these billions down to thousands of really expensive calculations, how do we handle that? So first, we have the hardest that we've done, and we have a team of really good solution architects and engineers that have built a direct cloud API. So that's how we've scaled up to 500,000 for a really long time. We are developing the technologies to not have to use the direct cloud API explicitly, but for a lot of our massive scale, we're still using that. What we're moving to right now is an auto-scaling queue system. As much stuff as, I shouldn't say we're moving to, as much stuff as we can handle there, we will. Slurm is our preferred queuing system, although we support many. It has some limitations in somewhere in the order of a couple 100,000 compute nodes, but reaching that and taking advantage of it, there's a bunch of other legacy stuff that you're reliant on. That said, it's extremely easy for some of our older applications to run on because they were built to run on clusters before the cloud existed. The future that we're building towards right now, is using Kubernetes and GKE specifically. I'll go on the next slide. I'll kind of go into the architecture details there, but we would really like to see a world where we can think about things in GKE clusters instead of having to manually do Cloud API. And we're hoping GKE will evolve to the place where it can even displace queuing systems for a lot of those tasks. The GKE setup that I discussed a second ago. So the way we're typically doing that now is we're using a Postgres server. Now I should mention that when we get to the point of really crazy compute, we'll almost certainly replace that Postgres server with BigQuery. Right now it's just easy to deploy. It also allows us to deploy it in-house if a customer were to have in-house requirements. The Postgres server and the Kubernetes spinner bring up nodes. They hit the Kubernetes API server to bring up nodes. We scale up to as many, I forget the Kubernetes term. We scale up to as many Kubernetes nodes as we can. And then as that work is finished, it'll kind of give status back to the Postgres server, maybe some very small number of calculation numbers, and it'll deposit the files into buckets. So the buckets will be used for both input and output, highly scalable data storage. That's actually one really big advantage over traditional HPC, which frequently still relies on things like NFS, is the buckets are just really high quality scalable data storage and cheap. So I wanted to stop a second and say, what exactly are those GPUs doing? When we're using 10,000 GPUs, I've talked about physics simulation, but what does that look like? And also, I just really love these videos. I think a lot of people think they look cool, so I figured I'd show one of these. So what this is right here is it's free energy perturbation calculation. I won't explain exactly what that means, but basically that's a drug molecule you're seeing with waters hovering around it inside a human protein. And what we're trying to figure out is if we take a drug molecule where we already know its efficacy and we make a small perturbation to it, will it be more effective as a drug molecule? Will it bind better to the protein? This calculation requires, here it says 8 hours of GPU time. I think the video is a few hours old and we have it down to something like 2 to 4 hours of GPU time, thanks to a lot to NVIDIA for speeding up their GPUs. But it takes hours on a GPU to do this simulation for a single ligand. And again, go back to that 10 to the 60th drug space. You can see how infinite the compute would be to calculate this for all available ligands. So simulating just that simple motion, those motion mechanics, is quite expensive, and we do what we can to reduce it when we can. So that says that's how we keep one GPU busy for that long. But coming up with ideas, how do we come up with so many ideas to keep all those GPUs busy? Well, it might kind of look like motion because it's so blurred, but what this actually is it's thousands and thousands of drug molecules superpositioned on top of each other, preserving the same core in the bottom. So those are all tiny, tiny changes up top. And we've enumerated many, many potential drug molecules that we think might be good. We've taken a lot of the human work out of individually drawing them by enumerating a bunch of different combinations. And once we've done that, now we say, okay, we have this big group of ones. Let's run some subset of these through the physics-based engine on the GPUs. So say we've enumerated 1 billion, like I was referencing. Let's filter it down. Let's run 1,000 through GPUs for several hours, maybe 10,000 for GPUs for several hours. And then when we get that data back, let's train an active learning algorithm. That active learning algorithm will then take a guess at another 1,000 values and just sporadically pick points that are either good or bad or maybe just interesting for other ideas and run the physics calculation on those. And that's how we kind of extend the applicability domain of active learning. Active learning is really good in this space for interpolation. When you have two things that are just clearly 2 steps apart from each other, active learning is pretty good at finding the point in between them. It's not perfect because sometimes there's surprise physics. We still always double check active learning, but it's really good. The thing that active learning isn't great at in this space is finding truly novel things. When you come up with new chemistry that is just going to totally change the way the physics in the protein works, active learning just can't guess that because it's never seen that physics in existence before with the protein. So that's where, for extrapolation, you really have to constantly be running more physics-based calculations. So once we've generated all of that data, what do we actually do with it? let's step back to the beginning of the drug discovery project. And a biologist has discovered a protein that they're pretty sure is strongly related to a disease. So they've done some lab experiments, and if they knock out that protein, the disease goes away, in the lab, not in a human being, maybe in a mouse. So let's kick off a project trying to find molecules that could knock out that protein, and maybe those molecules could end up being drugs. How do we actually do that? How much data is involved? And that's what the slide's talking about here. We probably want to start with something on the order of 10 billion new ideas. We want to do either high throughput screening in the lab, where they have some special machines that spend a bunch of fragments through, allow you to get some really inaccurate coarse data just to start off of. Or if you're pretty confident with what you have on the computer, it's called an X-ray crystal structure, 3D data of the protein, you can start to do virtual screening with just the computer itself, without having to go to the lab. People kind of go back and forth on these two things. They're both fairly inaccurate, but they find you the starting places you need to proceed. But the problem is, say you've calculated these 20 properties for 10 billion things. What do you actually do with this? Keep in mind, this is not a computer scientist. They're usually not even data scientists. This is just a chemist that has picked up computers over the years as they've had to. And we're talking about them having to deal with parsing, up to trillions of data points. So this is where GCP's BigQuery has really been a game changer for us. We're able to set it up so that our backends can deposit the most important data directly into BigQuery, and people who aren't necessarily data scientists are able to interact with that data. So BigQuery has given us a lot of great things. So in this example that you're seeing, we have loaded very easily 1.2 billion molecules. You can see the number of rows here, and a bunch of data about it into BigQuery. Now, normally, if we were doing this, these terabytes of data would require a non-trivial IT task. They'd have to set up a database for the user. The user would have to figure out how to interact with it. We'd have to pick a UI that the user can easily figure out, and it wouldn't scale very well unless we really were committed to making this great. But with BigQuery, as you just saw, you can execute this really simple query, this basic 5 drug-like properties query, and it will happen almost immediately in just a few seconds. So the scalability is like, we care more about the output of this than anything else we could care about from a database perspective. And it doesn't require very much for the chemist to get going on it. So it really has allowed us to think about data in a totally different way. This example shows how complex we can get. The first one was very simple, but this example shows us again, with 1.2 billion, we've created this really complex query, and what we're really trying to do is sort things into different classes. So we've loaded 1.2 billion new molecules in with all of their data, and now we have this query that allows us to sort those into different things. So you can see here, we've been able, in just 5.2 seconds, we've sorted 1.2 billion molecules into different classes of molecules, whether they're drug-like or fragments or things like that. The ability to do that essentially immediately is just an absolute game changer for our data scientists. So kind of the last really big GCP feature for us is not nearly as fashionable as machine learning or large-scale HPC or even big data. Honestly, it matters a lot for us, especially from the management, IT side, security side. We just love the project layout of GCP. The isolation is very, very natural. The user access management is great. It gives you a lot of options, sometimes too many options, but it's better to have too many than too few. And especially if you're a corporate G Suite user, it's just like trivial to move people into a GCP type cloud interface. I personally find the interface wonderful, both the web UI and the command line tools. I don't get enough time to do purely technical stuff anymore. So if the interface is really easy to learn, I'm much more likely to use the tool. And I find both interfaces to be really, really strong. I'm not going to give a bunch of examples here of the interface. People can play with it themselves, but I did want to highlight that because that was a really big part of the reason why we chose GCP, just the ease of use. It's enjoyable both for like naive users and the DevOps experts. So to kind of give the summary here, Schrodinger's computational chemistry platform can speed up drug discovery, and sometimes that's actually directly correlated to computational throughput. Sometimes it's blocked by lab work and things like that, but there are significant parts of a project runtime where if I just have the ability to run more computations at once, I will speed up how fast the drug goes to market. And it can literally be day for day. So if something used to take 10 days and it takes 2 days, that means you get to that clinical stage 8 days faster, and it's 8 days faster that drug gets to market. It's pretty amazing when you think about being able to speed up tasks like that. And the cloud has helped a lot. GCP, specifically for all the reasons I've listed today, makes our discovery efforts faster. It makes it more flexible. And that flexibility directly translates into getting things done faster. BigQuery allows our scientists to store and analyze data. That's something that it's kind of funny we're talking about in the same sentence because it seems like a totally separate tool, but the way it integrates so well with everything GCP is doing and how we can reach it from all of our compute nodes trivially, it has great access controls and all of that. That integrated nature of BigQuery really has been a game changer when we're analyzing the big data we generate. So all in summation, these tools come together to make GCP just a complete solution for Schrodinger that sped us up and we've really invested in. And then on that note, I'm going to go to the cool video. I love to show these. Again, I mentioned earlier, I think people just like to think about how molecules work inside your body, even if you're not a scientist. And we've done a pretty good job at making these things pretty. So here's a video that's a few years old that I'll kind of end the presentation on. And I will end it on that note. Thank you very much for watching today. I hope I didn't bore you too much with science. There's a ton of people I should thank, by the way. A bunch of our solution architects helped this out, and obviously the whole company built most of the software. Thank you very much.